All right, we are now going to move on to more complicated situations where multiple characters are different between the parents, and the next step is to look at a dihybrid cross. So a dihybrid cross requires the analysis of two characters, and it is assumed that the parents differ from at least two characters. And so this generates unique ratios in the F2 generation. And so as an example, we'll turn back to the peas, and we will consider plants that have round green peas, seeds, and wrinkled yellow seeds. So again, there's two characters in this case, the color of the seed and the shape of the seed, and each of those characters has two traits. So the color can be green or yellow, the shape can be smooth or wrinkled. And so we have chosen parents whose phenotype, if we contrast it to the second parent, differs at two different characters. So that's one of the necessities of having a dihybrid cross. The assumption is that there are two characters that are different between the two parents. Again, it is assumed that the parents breed true, and so they are expected to be homozygous. And we simply have filled in the dominant recessive relationship from the earlier chapter, from the earlier part of the chapter. So this individual is big R, little y, and this individual is little r, big Y. We do a cross in between these two parents, and so the first thing that you need to do now is to figure out what are the gametes that can be produced by this individual. And the answer is that this individual can only produce one type of gamete. It has to produce a gamete that's big R, little y. This individual has to produce a gamete that's little r, big Y. And that's part of the definition of someone that breeds true. If it was able to create multiple different types of gametes, then it would be able to have multiple types of offspring. So now we cross these two individuals together, and we create a heterozygote individual that is big R, big Y. And so this individual has a phenotype that is different from either of the parents. So it shares the yellow color with its second parent, and it shares the round shape with the first parent. But this phenotype is unique because a phenotype is a composite of all of the different traits. And so the way we figured that out was just based upon dominance recessive relationship. So we know that yellow is dominant to green, and we know that round is dominant to wrinkled. And so we use that information to figure out the phenotype. And so now we intercross these individuals so that they fertilize, they self-fertilize. And so now we look in the F2 generation to see what we see. And so when Mendel did this, he found four different phenotypes. So even though these parents all had the same phenotype, they were able to produce individuals that had four different phenotypes. So two of those phenotypes were the same as the parents, the round green, the wrinkled yellow. One of those phenotypes was the same as the F1 parents, that's the round yellow. And now we see a brand new phenotype that hasn't been seen before, which is the wrinkled green. So we see a total of four different phenotypes from this cross. And if we take the amount of individuals that have each of these phenotypes, we can find a ratio of approximately 9 to 3 to 3 to 1. So this is a very famous ratio that you should memorize and know. In a dihybrid cross, you see four different phenotypes, and they're at the ratio of 9 to 3 to 3 to 1. And so one of the things you'll notice is that if you look at the dominance recessive relationship, the dominant traits are the most common. So yellow is dominant, round is dominant, and so the yellow round is the most common, and then the least common is the recessive of both of the characters. So how do you figure out what the expected ratios are? So you can just kind of follow Mendel's approach very straightforwardly. And so here, unfortunately, the naming scheme of they come from actually different chapter books, so the naming scheme of the genes is different, but you should be able to follow this, I think, pretty straightforwardly. So again, we have a gene that controls the color, G, and the gene that controls the shape, and the F1 individuals are heterozygotes. And so now it's important to note, if we look at these individuals, we can calculate how many unique gametes can be formed. And so the answer is 4. So these are the 4 possible gametes that can be produced by this F1. It can produce the big G, and it can combine that with the big W. It can produce a big G combined with a little w. It can take the little g and combine it with the big W, or it can take the little g and combine it with the little w. And because this is an intercross, both parents share the same genotype, so the same is also true of the second parent. And so all we've done is listed the 4 types of gametes that can be produced by each parent, and then the inside of the Punnett square is what happens when you combine these gametes. So big G, big W, that was just taken by combining this GW and this one right here together. And we can do that for the entire thing. And so not only have we listed the genotype in this Punnett square, but we've also listed the phenotype that's expected to occur from any of the individuals that have this particular combination. And so you'll notice that there's 16 total squares within the Punnett square. And so this is kind of where the 9 to 3 to 3 to 1 ratio comes from. Because if you add 9 plus 3 plus 3 plus 1 together, you get a total of 16. And so all we've done down here is just combine. So you'll notice that some of these have the same genotype. So this and this are both big G, big W, little w. And so there's kind of 2 ways you can have that combination, depending upon which parent, both parents can provide you with the different gametes that allow that. And so, some of them have more. So this, right here, there's 4 different ways you can combine gametes to get the heterozygote combinations. And in some cases, there's only 1. So the only way you can get this combination is if both parents have the same. And so this table right here just lists what happens if we group all of these together by their genotype. So 1/16 of these will have big G, big W, big W. 4/16 will have this, the heterozygote, et cetera, et cetera. So if you just go through this, you will see like all the different possible genotypes that can occur. We can further group these by genotype. So these 4 right here, even though, you can group them by phenotype. So even though these 4 different offspring have different genotypes, they all actually share the same phenotype, which is yellow and round. And so by adding these up together, 1 plus 2 plus 2 plus 4, we get 9/16. And so 9/16 of them are expected to share the same yellow and round phenotype. And then so on and so forth we can do for the rest. And this is where the 9 to 3 to 3 to 1 ratio comes from. Essentially because of this independent distribution and kind of this equal probability of forming each of the different gametes. So if you simply multiply 4 times 4, you get 16, and then by grouping these together, by going through the exercise of defining all of the genotypes that would occur and then grouping them together, we can get this ratio. You can also move on and do something like a trihybrid cross. And so you've simply added another trait. We've added another character to that. And so now the parents are different not only at 2 different characters, but actually a 3rd character as well. And so if we do this cross, we would cross the parents together. We would get F1s. The F1s would all be heterozygote and identical. But then if we intercross these, we would get a much larger set of phenotypes that we observe. So we'd actually get 8 different phenotypes. And if we listed them all out, the ratio would be 27, 9 to 9 to 9 to 3 to 3 to 3 to 1. So 27, that's the 3 dominant traits, would have the dominant traits. So 27 out of 64 would have the dominant traits. 9, so for the categories that are 9 of 64, so they would have 2 dominant traits, and there's 3 different combinations that you can have for that, so there's 3 different categories. The 3 out of 64, they would have 1 dominant trait and 2 recessive traits, and there's 3 combinations you can have, and then there'd be 1 out of 64 chance of having a recessive combination of all. And so this is just kind of like a schematic of this. So we have, in this case, one individual that has all dominant traits, one individual that has all recessive traits. They breed true, they're homozygous. Each of them can produce a single gamete. So the only gamete this individual can produce is big A, big A, big B, big C. This individual can only produce a little a, little b, little c. The F1s are heterozygote by crossing these two gametes together, and they're all genotypically identical. And then if we look at the types of gametes that this individual can produce, there's actually 8 different combinations. So the big A can be paired with the big B and the big C. The big A can be paired with the big B and middle C, and so forth. So if we list all of those out, there's 8 total different gametes that can be produced. And so if we look back here, the reason there's 64 is because of that. There's 64 boxes because there's 8 gametes produced by parent 1, there's 8 gametes produced by parent 2, and then when we multiply those together, there's 64 total boxes. So think about that logic when you're thinking, if you ever wanted to do even more, how many, what would an expected ratio be if you did a cross that was, one more advanced where you had 4 different characters. How many gametes would be produced, and then if you multiply that together, that kind of gives you like the basics of the ratio that you'd be looking for. And so again, the reason that gametes, all 4 of those gametes are equally possible, this comes from our knowledge of meiosis that we covered in the previous chapter. So we have a heterozygote individual that is big G, big W, little w, and we said that all 4 of these combinations could be produced. So, we can match all of them together. And this simply comes from how the randomness of how homologs separate during meiosis. And so the reason you see independent assortment is often because genes are located on different chromosomes. So the gene that controls seed color is on a different chromosome as the gene that controlled seed shape. And so when you look at meiosis, this is when these homologs are actually separated out into the gametes. And so we can see a gamete, can have either the big G or the little g, just depending on how exactly it was separated. But if we looked at the coupling between the 2 genes, both of these couplings are possible. So just kind of randomly, each meiosis is randomly determined. Does the blue couple with the blue chromosome 1 couple with the orange chromosome 2, or do they couple together? And that coupling during meiosis 1 actually determines what that gamete genotype is going to be. And so since all 4 of these possibilities are allowed, since it's completely independent, which homologous chromosome goes with which homologous chromosome, then we know that there's kind of a 1/4 equal possibility for forming all of those gametes.